really like it.
Are you
All yours.
Okay. Good morning, gentlemen. My name is Michael, and I am here to talk to you today about self-renewal of embryonic stem cell-derived progenitors by organ-matched mesenchyme. So we're going to unpackage that phrasing. The article is by Julie Snedden, Mal Gorzada Borowiak, and Douglas A. Melton. They all work at the Harvard Institute for Regenerative Medicine. This was published in late 2012, so it's a recent article. So first, I'm going to explain what stepwise differentiation is and where the older frontier of embryonic stem cells research really was in late 2012. So stepwise differentiation is actually largely developed by Borowiak, Snedden, and the other Melton, who wrote this article. But currently before this article, what they could do is they could take an embryonic stem cell, and if they had X number of stem cells, they could lead them into becoming whatever type of cell that they wanted. So in this case, we're focusing on the pancreatic lineage because there's clear applications for diabetes and other organ systems. But let's say that you have the embryonic stem cell that's marked by ARCAMR4 binding protein. You can use certain chemical factors to turn it into definitive endoderm, which is indicated by the SRY box 17 containing gene. Then you can use more factors to turn it into pancreatic progenitors and endocrine progenitors, and finally an insulin secreting beta cell. So these are what also called islets, what your pancreas uses to break down glucose in your blood. So just remember that moving forward. The two that they chose to analyze in this experiment were definitive endoderm and endocrine progenitors, because one is relatively close to the beginning of the process and one is close to the end. So it's useful for analyzing their applications and how they work once you put them into biological systems. So the big problem with this, though, is that you're limited by how many embryonic stem cells you have at the very beginning. So if you have 100,000, you'll end up with about a 15% yield of beta cells, but you're limited by how many you start with, because at each step you're going to lose a couple with stepwise differentiation. So they wanted to know, how do we renew these cells? And there's a larger image of what stepwise differentiation looks like. And each of these is a transcription factor, so they can test for these proteins in these cells, and once they see a high concentration of SRY box containing gene 17, then they know it's definitive endoderm. Or if they see a C-peptide, they know that it's a beta cell. So that's what those indicators mean. All right, so they said the big question was, how can we amplify progenitor cells? These are all the questions, and you can read through them briefly, but we're going to walk through them one by one. The very first question is, which tissue can we place definitive endoderm and an endocrine progenitor on so that they amplify greatly, so that they self-renew? So they took, mouse embryonic stem cells and they used green fluorescent protein indicators, reporters. Then they separated them using fluorescence activated cell sorting. They took definitive endoderm and endocrine progenitors, and then they co-cultured them for 6 days on mesenchymal cells, endothelial cells, fibroblasts, and extracellular matrices. So mesenchyme is one of the 3 embryonic tissues. Endothelial cells is just sort of like a generic tissue, and then the latter two are just even more generic cellular environment, trying to simulate that. But whenever you see 6 days, that's exactly one passage. So they're putting these cells, these 2 types of cells, on each of these tissues for one passage to see how much they can renew. And then here's just a detail of they had to find durable lineages that could be passaged over and over and over for experimental purposes. And here are the sources for mesenchyme 1, 2, 3, and 4. Okay, so what they did see, to answer this question, which tissues can regenerate, is for the definitive endoderm cells, they saw a 9.7 and a 12.1-fold increase over one passage on mesenchyme 1 and 2. So these first 2 samples. Then for the endocrine progenitors, they saw a 14 and a 13-fold increase on mesenchyme 3 and 4, or the other 2 samples. So for all the other tissue types, endothelial, extracellular matrices, and fibroblasts, there wasn't any real proliferation pattern, or you never saw any real self-renewal. So the conclusion is that mesenchyme is the most useful tissue for amplifying these two cell types, and we'll talk more specifically about how those tissues work on others. So here you can see the results is for extracellular matrices, endothelial cells, other types of mesenchyme, and then you see that for endocrine progenitors, these blue bars, they're extremely high fold increase. Then on these two, for definitive endoderm, there's an extremely high fold increase. But if you look at definitive endoderm on 3 and 4, it's just along with the rest. So it's highly specific. All right, and they said, our first answer is, our first question has been answered. We know what tissue will work, but what is actually the underlying factor to this enormous growth? And they wanted to see, they tested it for 4 different things: selective survival, preferential attachment, induction of cells, and a decrease in cellular death. So we'll talk first about selective survival. They wanted to know, when we put these cells on specific tissues, are they surviving at the expense of other cells? Is there a selective survival effect that's happening? And as this indicates, when you co-culture them on mesenchyme 1 and 2, there's not an exponential growth that's usually indicated by selective survival or the natural selective process. And then, as you see here, wherever there are just a generic assortment of cells, there's also a high percentage of the definitive endoderm cells, and there's plenty of cellular death. So they're not beating the other cells, they're just proliferating enormously. And those are the lab procedures that you can read for yourself. And then phosphohistone H3 staining indicates cellular death, I believe. Okay, so the next question was, when you put the cells on mesenchyme, do they preferentially attach to the tissue? So is there an initial increase that just happens and more stick to the tissue? So they took fluorescence activated cell sorting purified definitive endoderm, plated them onto different mesenchyme samples: fibroblasts, irradiated mouse embryonic fibroblasts, IMEFs, epithelial tissues, and extracellular matrices, and then they checked the numbers 1 day later. As you can see here, this chart indicates the attachment, the number of cells that had attached to each tissue. So for mesenchyme, all the mesenchyme samples, they're one and the same with the IMEFs, the 804G cells, etc. They're all around like 6,000 to 9,000 number of definitive endoderm cells that attached. All right. The next question was, when you put it on mesenchyme, does this tissue actually cause more of the cells in our sample to become definitive endoderm cells? And then it has that effect. It actually changes the genetic, behavior. It changes the genetic state and the differentiated state of the cells that we're dealing with. So if we put generic samples onto mesenchyme, will they all of a sudden become definitive endoderm? And that's why they're able to grow so much. So they put both. I'll move on. They would, from the fluorescence activated cell sorting, they took the cells that were not definitive endoderm and then co-cultured them on mesenchyme and plenty of other tissues. Then they also took just generic embryonic stem cells and co-cultured them on these tissues. And they wanted to see, when you put them on mesenchyme, does it cause plenty of more cells to become these definitive endoderm that we've taken out of the process? And that does not happen. So when you take cells that were not definitive endoderm, there's not a lot of growth. So you have to hand mesenchyme the ball for them to run with it. They're not going to make something happen that you didn't produce initially. And then the last question is there a decrease in cellular death? And anyone that's taken AP Bio knows that caspase is a protein that indicates apoptosis. So apoptosis is cell death, cellular death. And they checked for the levels of the protein caspase in each of the samples. So when they co-cultured it with mesenchyme, NHDF, irradiated mice embryonic fibroblasts, etc., they checked for the level of this protein at different stages, and there's really no difference for each stage of the level of apoptosis. So there's not even a decrease in cellular death. There's just more being produced, and therefore more dying. Okay, so that answers those questions. There are plenty of factors that we just ruled out through experiments that show that it's nothing that simple. The next is, okay, we've realized that obviously it's a complex process. The cellular environment is hard to completely understand, but do the tissues affect all cells the same way? So is mesenchyme our go-to tissue that if we want to proliferate a progenitor, it's just going to work? So they put, well, first off, you can look at the initial reaction, or not the initial reaction, the initial trial, where they put definitive endoderm and endocrine progenitors on different mesenchyme samples. And as you can see, definitive endoderm doesn't proliferate on mesenchyme, on mesenchyme 3 and 4, and pancreatic endocrine progenitors do not proliferate on 1 and 2. So it's specific even depending on the cell type or the mesenchyme type. But if you also take embryonic stem cells marked by the octamer4 binding protein and put them on mesenchyme 1, 2, IMEFs, and other cells, they don't even prefer mesenchyme. So in this case, irradiated mice embryonic fibroblasts cause enormous cellular growth. So not only does it depend on the stage of the cell, but it also depends on the cellular tissue and the specific lineage that you're talking about. So it's an extremely complex process of why these cells are responding to mesenchyme, different sources of mesenchyme in different ways. The fourth question was, how can we trace a specific signaling pathway or a chemical occurrence that we can trace in a cellular environment? And is it due to one of these pathways that we can mark with chemical indicators? So they added all of these different chemicals that were supposed to spur what was happening and mimic the growth that happens on mesenchyme, the co-culture with mesenchyme. And as you can see, they took definitive endoderm cells and co-cultured them on extracellular matrices and IMEFs as sort of a generic cellular environment as somewhat a control. And then they added all these different signaling factors at different concentrations. And after one passage, they determined the fold change. So here's our fold change that we've been talking about. It's around 9% when it's averaged out. And here are the rest of what the signaling factors can add. So nothing, even the highest one, which is KGF and BMF protein, or BMP4, they were the greatest fold increase out of all the signaling factors, but they were only at about 4 or 5 fold increase. So that's about half the progress that we had before. It's not even close to what co-culture with mesenchyme does. So you can't attribute the growth to one signaling factor or one chemical pathway that happens in the cellular environment. It's much more complex. So there's no single signaling pathway that is promoted by co-culture with mesenchyme. Next, they said, well, we can't induce it by looking for chemical factors, but can we inhibit it by introducing different chemical factors? So they took all of these, they co-cultured it again on extracellular matrices and IMEFs, I believe, and they checked for SRY box containing gene 2 and FOXA2, which are both indicators of definitive endoderm. They were just sort of making sure that their results were accurate, and then they co-cultured them on mesenchyme 2, and then tried to use chemical inhibitors to reduce that process. So they introduced all these chemical factors that are supposed to inhibit cellular growth or block certain signaling pathways. But if you look at the positive control right here, the growth is at about this number, and then all the way across the board, it's roughly the same. So they can't even attack a certain chemical pathway and stop what happens when you co-culture on mesenchyme 2. So in this case of definitive endoderm, they can't attribute it to one certain factor. They can't say that it's selective survival, induction, preferential attachment, or decrease in cell death. You can't use one certain chemical signaling factor to aid the process, and you can't use any chemical inhibitor to stop the process. So it's the more experiments that they perform, the less that they understand about what's happening. But so now we've sort of thrown in the towel and saying we can't understand completely what's occurring right now and how the process works. But let's check these cells before and after we passage them. So before just using stepwise differentiation, we stopped at definitive endoderm and proliferated all those cells, and we also stopped at endocrine progenitors and proliferated all those cells. But before we stopped, or before we proliferated, what kind of genetic expression did they have? So on one side, on one axis, they plotted the genetic expression of all of their genes before, and then they plotted it after being passaged. And as you see, the slope for R squared is very close to 1, 0.92 and 0.96, depending on the mesenchyme sample that we're using. But the fact that it's so close after it's being passaged to the genetic expression that it had before it was passaged, they're functioning the same way in vitro, and they're sort of they're producing the same genetic expression, which is extremely important, that they don't lose their genetic identity or a lot of their expression. So also, as you see here, CDX2 and SOX2 are two transcription factors for later progenitors in the process. So right now, when we specialize, or when we're talking about definitive endoderm, we want it to stay definitive endoderm. We don't want it to lose its genetic identity and move forward in the stepwise differentiation process. But as you see, the percentage of cells that expressed these transcription factors before being passaged was about the same as after being passaged on these different tissue types. So when you put it on mesenchyme and it grows, it's not losing its identity, and it stays at definitive endoderm like we want it to. So it does not disrupt genetic expression. The next question was, does it disrupt further development? So we said it'll stay when we want it to stay, but will it go when we want it to go? Is it able to be passaged, or not passaged, but further differentiated through the stepwise differentiation process? So after being passaged 6 times, the definitive endoderm cells were differentiated into pancreatic progenitors, endocrine progenitors, and then those fully developed beta cells that we were talking about, which is sort of our end goal. As they saw, the unpassaged cells, the cells that weren't renewed at all, they just sort of started and then went down along the line, they had about that percentage yield of pancreatic progenitors, a roughly 20% yield of endocrine progenitors, and then an even lower yield of beta cells. So, and then when you compare that to the lineages that were passaged like 6 times on mesenchyme 1 and 2, it's about the same. So you can get 65 or 55% to be a pancreatic progenitor, and then it's right there when you passage the cells, you're getting the same percentage yield. But 55% of 1,000 cells is much smaller than 55% of 2 million cells. So being able to expand them and then differentiate them further means you have more cells to work with. So their developmental potential isn't lost. When you expand them to be definitive endoderm and you get this incredible fold increase, you're still going to get the same percentage, which is all important. Next, how do derivative cells from amplified definitive endoderm compare to those from unpassaged lineages in vitro? So in vitro just means that they're outside of a biological system right now. They're not being developed into, or they're not being inserted into the mice yet, or sort of asked to perform biological functions. We're just looking at them in a laboratory environment. So currently, you have a 0 to 15% yield of the beta cells that is sort of your end goal. So when you start with embryonic stem cells and through stepwise differentiation, you'll usually end up with 0 to 15% yield of those insulin-secreting cells that you really want. So they wanted to see, do we still get that yield with the definitive endoderm that's been passaged enormously? You still get around 15%. And then also, what they did is they checked this cell staining growth to check if we didn't passage the cells, what the development looks like, and if we do passage the cells and have them self-renew enormously, what does the development look like? So just through simple cell staining analysis and fluorescence-activated cell sorting, they're able to show that you have the same percentage of cells at each stage, which is also supported empirically a little bit later on. So they both end up with a relatively small yield of these beta cells, which are the green ones. that they used to using GFP reporters. But so they're both relatively unsuccessful in getting beta cells in vitro. But then they wanted to ask, what about when we start talking about inserting them into a biological system, so in vivo? Usually what people do now, wait, that's not on this slide actually. So so they what they do is they'll take they'll develop them all the way to endocrine progenitors, which is NGN3, the one that we were talking about earlier. And they'll get it to the step right before being a C-peptide cell, and then they'll insert them and let them differentiate into the beta cells in vivo. So once they're in the mice, to have a much larger percentage yield, because that's more successful. So they inserted all of these cells into the beige mice and tested using enzyme-linked immunosorbent assay to check for the antibodies of C-peptides. And as you see, when you put the unpassaged cells into the mice and the passaged cells into the mice, you get close to the same yield. And in most cases, it's actually better that they're passaged because they produce higher percentages of each step cell, or each differentiated cell. So you're getting a higher percentage in this case of the SRY box-containing gene expressing cells, the PDX1 expressing cells, and then the neurogene-3 expressing cells. But the final question, the reason we've been talking about all of this is because we want to see if the beta cells are going to respond in a biological environment, if they can actually secrete insulin in a glucose-responsive manner. So if the cells don't actually work, then this is all for naught. So what they do is they put human islets or just pure pancreatic insulin-secreting cells as a positive control into mice. They also put negative controls like a phosphate buffered saline solution and mesenchyme into these biological systems, in addition to the lineages that had been passaged over time on our mesenchyme. So the animals were fasted for 16 hours, then tested with the glucose injection shot to track their peak blood glucose level. The negative controls had a peak blood glucose of about 450 milligrams per deciliter, and then the positive control, the islets, were right at 300 milligrams per deciliter. And we'll move ahead for 1 second. So here's the negative control, the phosphate buffered saline solution, and the pure mesenchyme. They didn't help much, and the peak blood glucose is pretty high. But the purple line indicates the positive control, and as you can see, the passage cells, the blue line, is relatively close at minimizing that peak blood glucose. So it does break down glucose responsibly, or it does produce cells that break down glucose responsibly. And then they also wanted to check 45 minutes after the glucose injection for C-peptide levels. Again, C-peptide is the transcription factor for the beta cells that we want to analyze. So they checked C-peptide levels in the plasma of these mice afterward. And as you can see, for the negative controls, it's incredibly low. For the positive controls, it's relatively high. And then these are close enough to the positive controls that you see that they're useful. Also, the p-values are incredibly low, so you know that it's relatively consistent with our hypothesis. So, also considering these numbers, they're sort of subject to fluctuations based on trying to deliver a uniform amount of cells and grafting the cells, hoping that they survive. So when you start putting the cells into biological systems, it's not as exact as you hope it could be, and some of the numbers might be a little bit off. But you do see a generic trend that the negative controls were not successful at all. The positive controls were as successful as you would expect, and our passage lineages were relatively close to the positive controls. So our conclusion is that the derivatives of expanded progenitors are biologically responsive and can function well implanted into living systems. So our final answers to each of the 10 questions we had at the beginning is, first, mesenchyme is extremely successful in proliferating the specific progenitors that we were talking about, definitive endoderm and neurogene-3. They accounted that they accounted for the facts that growth maybe could have been caused by induction, proliferation, attachment, or an increase in apoptosis, a decrease in apoptosis and cell death. But they found that that was not the case. It's not caused by any of those factors. They can't show a single chemical factor that explains it or can impair this proliferation. And also, it's highly specific to various tissue types, to the cell type, where it is in stepwise differentiation, what stage it occurs. It does not impair the genetic expression in vivo. It does not impair its developmental potential trying to move forward in the stepwise differentiation process. And it also responds very similarly to unpassaged lineages in vitro. So they have about the same yield. And then once you implant them in vivo into a biological system, they develop very well. And as we saw, often better than the unpassaged lineages. Our final and biggest answer is that the final beta cells that are produced by the renewed mesenchyme, or by the renewed tissue on mesenchyme, does secrete insulin in a glucose-responsive manner. So, what does this mean for the scientific community moving forward? First, they're not limited anymore by how many embryonic stem cells they have at the beginning of their experimental process. Before, you had to have, let's say, 1 million embryonic stem cells and just hope you'd get a smaller yield at the end, just by chance. You'd have to go along the stepwise differentiation process, but you were limited at the very beginning by how many you had. Now, you can stop in the middle, enormously proliferate these cells and have them self-renew on different tissue types, and then move forward, and then you end up with a much greater yield at the end, which is incredibly significant. So they're going to be more easily able to perform research on embryonic stem cells, and obviously the source of embryonic stem cells is a source of controversy, so you can reduce that. But when you also talk about regenerative medicine, being able to take cells and put them successfully into a biological system where they operate the way that they're supposed to, and they can perform functions that often damage cells, or people that have cells that are disrupted by certain illnesses like diabetes, if they can do that job successfully, it has incredible impacts, not only for the pancreatic lineage. You can talk about cardiac, kidney, intestinal, all these different types of cells that can help cure disease and other illnesses. So it's going to have a huge role in medicine, it's going to have a huge role in research, and it's going to have a huge role in science in general. So that's the end of my presentation.